Welcome to AP Biology. I'm Allison Kate, and today's lesson is going to be on photosynthesis. We're also going to be looking at Science Practice 3B, which is practicing stating the null and or alternative hypotheses, also being able to predict the results of an experiment. And we're going to apply that to Biology Lab Investigation 5, on photosynthesis. So don't forget your pen and paper, and let's go ahead and get started. Some things that you should be familiar with before we get rolling today are chloroplast structure, and hopefully you paid or tuned in to our lesson on cell structure and function. If not, I'll review a little bit about chloroplast structure. You should also be familiar with the floating leaf disc assay. This is the experiment that we're going to do in Lab Investigation 5. And the terms photosystem, pigment, electron transport chain, and proton gradient. So today, a lot of this material is dense, and I'm going to do my best to take you through it. By the end of today's and you'll be able to describe the photosynthetic processes that allow organisms to capture and store energy. You should also be able to explain how to capture energy from light and temperature or pH of the reaction enough. So if you're doing temperature, maybe you started out at 10 degrees C and you went all the way up to 50 degrees C. We know that for most enzymes, a temperature that is optimum would be body temperature, so around 37 degrees. So that could describe the results we saw. We saw a peak in the middle with low at the beginning. So if that was a low temperature, there wasn't enough kinetic energy to get that reaction going. And if it was at the high end, you could have denatured the enzyme with that high temperature, breaking the hydrogen bonds. And same thing with pH. You would want to have a range of pH. Enzymes do have an optimum range of pH, although there are some that work best at low pH, like the enzymes in your stomach, or at higher pH. So you need to make sure you have a sufficient range. You need to make sure you held the other experimental factors constant. So for example, if you're focusing in on a temperature variable, then you would hold the pH constant. If you're focusing in on a pH variable, you would want to hold the temperature constant, so that you need to have all of those things so that you're only focusing on your variable that you're testing. And then you need a control, right? The control group is what you compare your experimental results to. And for this particular experimental design, you would either have no enzyme or an inactive or denatured enzyme, or you would have no substrate. You need to, in your design, make sure that you have some way of verifying your results, that you have repeated trials, or that the results you get represent your average or mean data. And then finally, that hypothesis must be clearly stated. You can use an if-then format. You can make a prediction. You can say something like the enzyme activity will be greatest at 37 degrees Celsius, or the enzyme activity will be greatest at pH 7. Okay, so that's the scoring guidelines, and I hope you guys did well on that one. All right, so today's lesson about photosynthesis, here's what you need to know. And I'm actually going to give you a lot of details about photosynthesis, then we're going to apply it to the lab, and then we are actually going to practice with some FRQs. So first of all, overview. Organisms capture and store energy for use in biological processes. The statement from the CED, that's the course and exam description, is that photosynthesis captures energy from the sun and produces sugars. And so what you are looking at is an overall image, and it is situated in the chloroplast. So we'll get into the details of chloroplast structure in a minute. The top blue, water, and gray, carbon dioxide, represent the reactants of photosynthesis. And at the bottom, that red, oxygen molecule, and the green square that says sucrose represents the chemical energy that's produced. You can also see that there are two different types of reactions going on in the chloroplast, and they take place in different places, but they are connected, and that light is involved. Okay, so we're going to go through this in detail in the next several slides. All right, but before we get into the details of the process of photosynthesis, we want to get an evolutionary lens on this process. And the first thing you should understand is that photosynthesis first evolved in prokaryotic organisms. And there's lots of scientific evidence to support the claim that prokaryotic or cyanobacterial photosynthesis was responsible for the production of an oxygenated atmosphere. So we owe the fact that as aerobic organisms, the atmosphere has enough oxygen for us to breathe. So I have an image of cyanobacteria. It's on the left-hand side, and these are the organisms that built up the oxygen in Earth's atmosphere over many millions of years. The second thing is that prokaryotic photosynthetic pathways are the foundation of eukaryotic photosynthesis. So eukaryotic photosynthetic pathways evolved from these early prokaryotic photosynthetic pathways. And you can see in the image on the right, we have purple sulfur bacteria, and they have a prokaryotic photosynthetic pathway that uses a different pigment to capture energy. And you may have heard of this. It's called bacteriorhodopsin, as opposed to the chlorophyll pigments that we see in eukaryotic photosynthesis. So a revisit to the chloroplast, they are the site of photosynthesis. It is where solar energy is converted into chemical energy. And just remember that there's several membrane systems that create compartments. So this concept of compartmentalization, which we've talked about in previous lessons, is really important as we talk about this process. So there's an outer membrane and an inner membrane, and in the chloroplast, they're both smooth. There is a space between them called the intermembrane space. Then we have a third membrane system, and it is called a thylakoid membrane, and they are folded membranes. That name actually comes from a stack of coins, and they're stacked up into what is called a granum. And in this image, you can see that there are 4 grana. So there's lots and lots of thylakoid membranes that are folded up. Outside the grana is a dense solution called the stroma, and I've indicated here that this is the site of the light-independent reactions. I may date myself. I learned these as the light and dark reactions. So the light-independent reactions, formerly known as the dark reactions, can take place in the presence or absence of light. The thylakoid membranes is where the light reactions or light-dependent reactions take place. So we have some compartmentalization, some separation of these two parts of the photosynthetic process. So just keep that in mind, that these compartments are really important. And as I talk a little bit more about the different specific steps of this process, you can think about where they take place. Okay, so hang on. Okay, so photo, if you deconstruct this word, photo, light, synthesis means to make. Photosynthesis is actually composed of light reactions, which again take place in the thylakoid membranes, and the Calvin cycle, which is where we actually convert carbon dioxide into organic molecules. So we have photo and synthesis. And again, you're seeing that same image that shows those 2 reactants to build one 6-carbon sugar molecule. It is 6 carbon dioxides that are taken from the atmosphere and 6 water molecules that are taken from the environment, along with light, energy, and a way to capture it. We are able to produce a 6-carbon organic molecule and release those 6 oxygen molecules. And those are the oxygen molecules that go out into the atmosphere and make it possible for aerobic organisms to do cellular respiration. And tomorrow's lesson is on cellular respiration. And so you're going to see how these 2 energy processes are related. All right, so before we get going, I want to talk a little bit more about light. So we need to talk about the solar energy part of this. Light is just energy, and it's part of a larger electromagnetic spectrum of radiation. Shorter wavelengths, which are represented by that Greek letter lambda, have higher energy, whereas the longer wavelengths have lower energy. Visible light is the light that is able to be detected by the human eye. And as you look at that whole electromagnetic spectrum, you can see way down on the short, high energy end, we have gamma rays, and then down on the longer wavelength, lower energy, we have radio waves. Visible light is just a very small part of the entire electromagnetic spectrum. And the visible spectrum, you guys may know because you've seen rainbows or you have used prisms, so you can see the different colors of light. The shortest wavelength of visible light is around 380 nanometers, and it's down in the violet range. And then the longest wavelengths is around 750 nanometers, and those are in the red range. And you may have remembered or learned that the colors in this visible light are Roy G. Biv, and that's working backwards from long to short. So you have red, orange, yellow, green, blue, indigo, and violet. So those are the colors that are visible to the human eye. Now, when light strikes a chloroplast, three things can happen, right? It can be reflected, which because the pigment is green, it will reflect that green light, and that's why plants appear green to us. Light can be transmitted, which means it just passes straight through, or it can be absorbed, and the absorption of that light is what we need in photosynthesis. So we have special molecules that will absorb that light. By the way, you have molecules in your eyes, in your retina, that actually also absorb light that allows you to see. Same idea here. So let's talk about the photosynthetic pigments. They absorb different wavelengths of light, and the reason that we have multiple pigments is to expand that visible spectrum of light that plants are able to use to drive photosynthesis. So what you're looking at in that image is the absorption spectrum of three different photosynthetic pigments. We have chlorophylls, and there's two kinds, chlorophyll A, which in that image is the blue-green line, and you can see it has two peaks. It will absorb maximally at about 440 nanometers and again at 680 nanometers. And chlorophyll B is that green-yellow line. It's kind of an olive color versus the blue-green, and it absorbs at 470 nanometers and again at 700 nanometers. Notice, however, that where the two green lines are lowest is in that range of green light, and that tells you that they reflect green light. And then we have a third set of pigments, which are collectively known as the carotenoids. They are actually beta carotene, which is what gives carrots their orange color, and then xanthophylls, which are yellow pigments. And you can see they have some peaks down in the violet blue range. And notice that they drop off after that. They provide, again, an extension of the amount of light that can be absorbed, but they also are involved in photoprotection. And it's kind of like if you go out in the sun and you want protection from solar radiation so you don't get a sunburn, you put on sunscreen. And so you can think of the carotenoids as kind of the sunscreen of the photosynthetic pigments. Okay, so now we're going to get into the tough part. So we're going to talk about how the light-dependent reactions of photosynthesis and eukaryotes actually work. And let me just tell you that the whole point of these reactions is to capture that solar radiation, that solar energy, and convert it into some forms that the plants can use. And the forms that we're going to talk about are ATP, which is the universal energy carrier in cells, and you're going to get lots more instruction about that in tomorrow's lesson. And then a molecule called NADPH. And I'm just going to leave it at that. It's got a really long name, but we just know it as NADPH, and the P is for photosynthesis. Tomorrow, when you talk cell respiration, you're going to see a related molecule that does not have the P. And so the whole point of this is to produce the ATP and the NADPH that will be used in the Calvin cycle, which is the light independent part of the photosynthetic reactions that will actually generate our chemical sugar energy. Okay, so let's start off with the image, and I'm going to kind of talk you through it. This is an image of a photosystem. A photosystem has a collection of pigment molecules. In this case, they're the chlorophyll A molecules that we're illustrating here. And it's going to have a light harvesting complex and a reaction center and then an electron transport chain. And I want you to see that this is embedded in the thylakoid membrane, which is just a phospholipid bilayer that you should recognize. And I want you to notice that on top of this image out here is the stroma, and then down here is the thylakoid space. And this directionality, this compartmentalization is important to understand, like the how of this process. All right, so we have a photon of light. It's going to strike the thylakoid membrane, and then we have green pigments known as chlorophylls. Now, a little bit of chemistry here. Chlorophyll has at its core a magnesium atom, and the magnesium atom is going to actually lose its electrons. So that energy is going to, when it eventually gets transferred, you can see these black arrows, they're transferring all that energy amongst the chlorophyll molecules to a special pair of chlorophyll molecules that will actually, they get so excited they lose their electrons. And those electrons get boosted up to a higher electron acceptor, and this is called the primary electron acceptor, and that's right here. And those electrons are grabbed by that primary electron acceptor, and they are going to be passed down an electron transport chain. Okay, so now let's look at these two photosystems that work together. And my students are always confused when I tell them that the first part of this light harvesting reaction occurs in photosystem 2. And the reason for that is that it was discovered second. Photosystem 1 actually evolved first, but it comes second in the process. So what you see is they're very similar, but they have different reactive chlorophyll A molecules. So in this first image, and I'm going to show you this in more detail in the next slide, but in this image, light is absorbed by chlorophyll. And it gets transferred to that primary electron acceptor, which receives it, and then it's going to pass those electrons down an electron transport chain, which is a series of proteins and molecules that are embedded in that thylakoid membrane. And out as it transfers those electrons, it's also going to pump protons into the thylakoid space. Okay, let's back up. Where do the protons come from? So water, remember, water is part of the reactant. The first part of this reaction, light splits that water molecule, and that is called photolysis. That's actually what produces the oxygen byproduct. So plants want to get rid of that, which is a good deal for other animals and other oxygen breathing organisms, right? So that also, when we split water, we generate some hydrogen ions, and those hydrogen ions are going to be key in setting up a proton gradient between those two photosystems, and we're going to use that energy to make ATP. The second thing that happens is we're going to pass those electrons, but we're also going to add more energy to a second photosystem, which will pick up that energy and boost that up to a primary electron acceptor in photosystem 1. This time, rather than generating that proton gradient, these electrons are going to be transferred directly to some proteins in the thylakoid membrane, ultimately activating this enzyme, which is called NADP reductase. And that tells you what it's going to do. It's going to actually reduce NADP to NADPH. And that is how we get ATP and NADPH from the first part of these reactions. Those two molecules, the ATP and the NADPH, are going to be the energy that originated from solar energy that will be used to produce sugars in the next phase. I know that this is tough, so hang in there. So this is really important, and actually today's lesson, I think, is going to set the stage for tomorrow's lesson, because you're going to apply the same concept with a few different details, a few tweaks to the process of what happens in cellular respiration in a similar organelle, the mitochondrion. Okay, so that both the mitochondrion and the chloroplast use this proton gradient to generate ATP. So again, orient yourself. You're looking at the thylakoid membrane. The thylakoid space is in the middle, and you've got the stroma surrounding the thylakoid membrane. So let's go ahead and take a look as I kind of hit the highlights here. So light is absorbed by photosystem 2. Light also splits water. The electrons that come from the water molecule are going to be used to replace the ones that are lost when the chlorophyll A loses its that are ultimately passed down the electron transport chain. The oxygen diffuses out and is a byproduct of the process. The protons are actually going to be stored in the thylakoid space. So now we're going to move those electrons down the electron transport chain, and as we do that, we move more protons into the thylakoid space. Then that proton gradient, so we've got way more protons inside the thylakoid space than outside of it in the stroma, they're going to be used to generate the energy that powers the production of ATP. So if you look here, there's a very important part of this membrane proteins, and this is a special enzyme called ATP synthase, and this is the site of ATP production. And the way that ATP is produced is that ADP, adenosine triphosphate, gains a phosphate to form ATP, or adenosine triphosphate. And now my students always ask me two things. First of all, what is inorganic phosphate and where does it come from? So it is always shown as this P for the phosphate, and then I for inorganic. And it is just floating around in the cell. So it may be there from the reverse reaction of ATP being broken down back into ADP and phosphate. So it's there. And again, what is creating the ATP through the ATP synthase enzyme is that proton gradient. So the next thing that happens, or a continuation, those electrons from photosystem 1 are used to transfer directly to the enzyme NADP reductase, and that causes the NADP to be reduced to NADPH. So now we're going to take those two energy producing or energy molecules, ATP and NADPH, to the Calvin cycle. Okay, so you can go back and look at these and kind of go slow because that's a lot of information. All right, so now we're finally at the place where we're going to finally make some organic molecule. We're going to make some sugar. And so the energy that was captured in those light reactions that I just went over, how it's transferred to ATP and NADPH, those two energy molecules are going to power the production of carbohydrates from carbon dioxide in a cycle known as the Calvin cycle. And this occurs in the stroma of the chloroplast. And it actually is going to produce a 3-carbon sugar called glyceraldehyde 3-phosphate, or G3P for short. So I know the overall balanced equation of photosynthesis tells us we're going to make glucose, but it actually takes two turns of this Calvin cycle to get you that 6-carbon sugar. It occurs in 3 phases. They are the carbon fixation phase, reduction phase, and regeneration of something called RUBP. So I'm going to take you through those phases. But first I have a shout out. This cycle is named after Melvin Calvin, who elucidated the steps of this cycle using an algae called chlorella, and his work was done at UC Berkeley. So go Bears. All right, so let's take a look at carbon fixation. What the heck does that mean? Carbon dioxide from the atmosphere is brought into the plant and into the Calvin cycle in the stroma, and it needs to be fixed or converted into organic molecules. And there is an enzyme that does it. Its name is called ribisco. It stands for ribulose bisphosphate carboxylase, and that's a mouthful. I had trouble getting it out. So we just call it ribisco. And it turns out that ribisco is the most abundant enzyme in the plant. A full 15% of the plant's protein is this one enzyme. And what it does is that it grabs the CO2 from the atmosphere and combines it with a 5-carbon molecule from the cycle called RUBP. And there is a super short-lived intermediate, and then that breaks down into another chemical called 3-phosphoglycerate. And now we're going to take that energy from ATP and NADPH to reduce it. So we're going to add carbons to make that molecule larger. And we're going to do that till we create a glyceraldehyde 3P phosphate G3P molecule. So carbon fixation is the first step. It uses the enzyme ribisco, and it forms a organic molecule. That organic molecule is reduced using ATP and NADPH to form that 3-carbon G3P. And the final phase, since it's a cycle, is we have to regenerate that 5-carbon RUBP molecule so that it's ready to begin the cycle again. And again, it takes 2 cycles to create that 6-carbon carbohydrate. It's really important to note right here that this pathway first creates a 3-carbon sugar, and it's called the C3 pathway. There are variations on this pathway. One of them is called a C4 pathway, and you might know that it's going to start off producing a 4-carbon sugar. It uses a different enzyme. And then there is yet another variation called CAM pathway. Neither the C4 or CAM pathway are going to be discussed here. They are outside the scope of this topic 3.5. Okay, so just a review. During photosynthesis, chlorophylls absorb energy from light, boost electrons to a higher energy level in both photosystems 1 and 2. Photosystems 1 and 2 are embedded in the internal membranes of chloroplast and are connected by the transfer of higher energy electrons through the electron transport chain. When electrons are transferred between molecules in a sequence of reactions as they pass through the ETC, an electrochemical gradient of protons, or those hydrogen ions, is established across that internal membrane. The formation of the proton gradient is linked to the synthesis of ATP from ADP and inorganic phosphate via the enzyme ATP synthase. The energy captured in the light reactions and transferred to ATP and NADPH powers the production of carbohydrates from carbon dioxide in the Calvin cycle, which occurs in the stroma of the chloroplast. So that's your kind of your money slide. That is the review of those steps. And hopefully the lesson and taking you through some of those images helps you understand that summary. All right, so now I want to apply this to a lab, and you may have done this lab in your class, but this is the leaf disc assay. And just a note, you may see that word disc spelled differently, and you'll probably see it spelled differently in my presentation. I don't know the correct spelling. It's either D-I-S-C or D-I-S-K. So anyway, just don't get confused by that. So in this lab, the question we're asking is what factors affect the rate of photosynthesis in living leaves. In this lab, you have 2 objectives. You want to be able to design and conduct an experiment to explore the effect of different environmental variables on the rate of cellular photosynthesis. And then you're going to connect and apply many concepts, including the relationship between cell structure and function, like we just saw in the chloroplast, and the idea of those membrane systems and compartmentalization. We're also going to try to connect the concepts of strategies for capture, storage, and use of free energy, and then also diffusion of gases across cell membranes. And the gases, of course, are carbon dioxide and oxygen, and the physical laws pertaining to the properties and behaviors of those 2 gases. So I'm starting you off with an image. I've got a cross-section of a leaf, and there are 2 things that are important about this. The first one is that you see that there is a palisade layer, and there's lots of air spaces around it. There's also air spaces in what's called the spongy mesophyll. Those air spaces are going to be important to the design of this experiment. Also, I want you to see that those palisade layer is packed with chloroplasts. Those are the chloroplasts where the photosynthesis reactions are going to take place. This experiment uses light. You can actually do this outside if you have bright sunlight, or you can use a lamp. And syringes. And the syringes are important because we need to start this experiment by removing all of the carbon dioxide and oxygen that exists in those leaf cells. Okay, so let's take a look at this next slide. I'm going to show you, before we get into the data table and the graph, I want to talk a little bit more about how you get this set up. So you can see there are syringes. You could do this at home if you wanted to play with this. If you have never done this before, it's super, materials are super easy to get. You just need some leaves. In my class, we use spinach leaves, or you can go on a field trip and just pick leaves from your school or your backyard. And then you just need to have some way to make constant the size of the leaf disc that you're going to cut. So you can use a hole puncher, you could use a straw, and you just want to make sure that you have discs of same size. One other thing, you don't want to cut your leaf discs out of the veins because that is the vascular tissue and there's no chloroplast there. So you cut your discs out. We cut out 10 discs. We will put them in the syringe. And the reason that that's important is we're going to fill that up with a solution of water and baking soda, just baking soda like you have when you make your chocolate chip cookies. And that's going to provide the carbon dioxide, the carbon source for photosynthesis to take place. And you are going to get rid of all those gases inside the leaf disc by putting your thumb over the top of the syringe and pulling on the plunger. And what happens, you'll feel that pressure and all that. You'll see air bubbles, and all those air bubbles you want to get rid of. So you do that by pushing the fluid all the way to the top. And if you do this properly, all of your leaf discs will sink. Then what you do is you take them out of your syringe. Some people leave them in the syringe, but you take them out, put them in a beaker of your baking soda solution, and then you turn the light on. And what we're going to do first is just practice this technique by looking at light intensity. So I just threw together some sample data. And so the way that we do light intensity is we measure 0, 5, 10, and 15. The 0 actually is no light, but 5, 10, and 15 centimeters away from the light. So the beaker is that far away from the light source. And what we actually measure is the time it takes for half of those leaf discs to float. And you're probably thinking, well, how do those leaf discs float? Once photosynthesis happens, right, the cells start taking in the carbon dioxide and they start producing oxygen. When they produce oxygen, it infiltrates those air spaces and it causes those leaf discs to become buoyant. And so they begin to float. And so it is a measure of the net photosynthetic rate. And so what you can see here is that the higher the closer the light is to the source, the higher the light intensity, the faster the time is, the faster, but the shorter the time compared to the 10 centimeters takes a little bit longer, and the 15 centimeter takes longest. The no light we have indicated here because photosynthesis will never happen without light. And so in the class, we just let it go for 1500 seconds and then call it good if nothing has, none of the discs have risen. Okay. Okay, so the next thing is to have you guys design your own experiment. And this is the experimental organizer that I use. I have my students decide a possible variable, create a title, make a prediction, indicate what the independent variable is, how many levels of independent variable you're going to have, how many trials you're going to do, how you're going to measure it, and what your experimental constants are. So if you wanted to brainstorm some variables, you could investigate temperature, wavelength of light, C4 versus C3 plants, the size of the leaf disc, the amount of carbon dioxide, sun plants versus shade plants, leaf color, leaf size. Okay, so you go ahead and pick your variable. And this is an example from my class. This is from a group that looked at light color, and the way they did that is using different colored filters. So looks like they looked at red light, yellow light, green light, blue light, and then just regular old white light, unfiltered light. And as you look at their graph, you can see that the one that went the fastest is the lower bar, and that is all wavelengths of light. The one that took the longest is the green light, and that is because green light reflects rather than absorbs light. So there's not enough energy to drive photosynthesis. So my students would show their work by creating a claim that the photosynthetic rate would be lowest when the light color is green and highest when the light color is red. Their evidence is their graph, and their reasoning connects their evidence to the claim. Okay. Just one more and then I'll move on. This is a group that did temperature, and you can see that there is no data for low temperature because there's not enough kinetic energy to actually drive the photosynthetic process. And then at high temperature, the rate is actually lower, and that's because we perhaps denatured our enzymes. So we can see that at 25 degrees Celsius, that had the maximum photosynthetic rate. All right, let's get some practice. So my first practice question is looking at kind of the evolutionary lens of photosynthesis. And I've got fossils of a microscopic organism. They are found in rocks that were determined to be over 3.5 billion years old. And the question asks you to identify two types of evidence that would help answer the question of whether the organism was photosynthetic. Okay, so how would you know looking at these old rocks? So you would know if there was some type of presence in the fossil of photosynthetic membranes or some type of imprint of chloroplast. Another way, and probably a more realistic way, is that there would be high levels of oxygen gas or oxides. The rocks would be rusty in the surrounding rock. You might see evidence of photosynthetic pigments, or you might see a chemical analysis of the fossil showing the presence of molecules that are associated with photosynthesis. Another way you could do this would to do it backwards. If there was a lack of photosynthetic membrane or chloroplast, or a lack of that oxygen oxidation in the rock, that would suggest an anaerobic environment. All right, let's look at an actual photosynthesis one, not an evolutionary one. And this is a really good question. So there's a lot of information in the stem of the question, and I will read it to you, but I'd like you to read along with me. And then the data table is showing you the photosynthetic rates of 3 groups of shrubs that were grown under varying amounts of illumination. So this is like the light intensity. So we've got shrubs growing in full sun, shrubs growing in partial sun, and shrubs growing in the shade. And you can see all of their data with different intensities. So the intensity is the independent variable. So researchers tested the effect of light on the rate of photosynthesis by using a shrub growing under conditions that differed widely in the amount of variable light, but where the availability of water and soil nutrients is fairly constant. So we're holding water availability and nutrient availability constant. Under constant temperature, relative humidity, leaf surface area, so more constants, the researchers used increasing illumination, which is measured as the photosynthetic photon flux density, the number of photons of wavelengths between 400 and 700 nanometers per unit surface area and unit time, and determine the net photosynthetic rate. And that was measured by the amount of carbon dioxide fixed per unit surface area and unit time at each illumination. Okay, so that's a lot of information. So the first thing you're asked to do is to describe how plants absorb photons of light. So hopefully you could do that by saying that plants contain pigments such as chlorophylls that will absorb photons of light at certain wavelengths. And that's the visible light between that 400 and 700 nanometers. Next, you're going to be asked to construct an appropriately labeled graph to represent that data. And I got us started on the graph grid by indicating the independent variable. And the independent variable is the type of environment, right? So it was grown in full sun, partial sun, and shade. And then I've got my units here of the photosynthetic photon flux density, which is different in each of those conditions. And then my dependent variable, what was measured, which was how much carbon dioxide was fixed, and that's the net leaf photosynthesis in micromoles per carbon dioxide per meter squared per second. And so the scoring guidelines would tell you that you would get full points if you had your correct axis labeling, as I just indicated, that you've got your correct scaling and units, and that you have your correctly plotted lines. And in this case, because our independent variable is categories, we're going to use a bar graph. And so if I graph that data, I'm going to get full sun, partial sun, and shade. And you can clearly see from this graph that the full sun has the highest amount of net leaf photosynthesis and the amount of carbon dioxide that was converted into carbon. Okay. So if you look at that top line, this is going to become important in the next part of this question. Okay. So based on the data, identify the shrubs and data point where the greatest flow of electrons through the chloroplast electron transport chain most likely occurs. So we know it's going to be the shrubs in full sun, and that data point that I highlighted in red is at 1500 micromoles per meter squared per second. And finally, predict the effect of growing shrubs growing in one area if they are tested with a compound that blocks electron flow through the electron transport chains of chloroplasts, and then provide reasoning to justify that prediction. So we know anything that's going to block the electron flow is going to prevent enough energy being produced to drive the production of sugar, so the plants are going to die. If they can't make their own food, they are not going to fare well. So your prediction is that the shrubs will die, they'll eventually die, they'll not be able to make sugars. And the justification for that is that blocking that electron transport chain is going to prevent the formation of the proton gradient, which is needed to produce ATP, and that ATP is needed to produce sugars. So that's a really good practice question. So go back and watch this and look at it. So just a quick check for understanding. Remember, our skill was to state the null or alternative hypothesis and predict the results of the experiment. So let's take a look at that first photo leaf disc experiment, where we looked at the variable of color or wavelength of light. So a null hypothesis might be the color of light will have no effect on the rate of photosynthesis. An alternative hypothesis might be that the photosynthetic rate will be lowest when the light color is green and highest when it's red. And to predict the results, the photosynthetic rate would be greatest in red. Since chlorophyll absorbs in that red range, it will be lowest in green because chlorophyll reflects green light and will lack the energy necessary to drive the reactions of photosynthesis. One more quick skill practice, this time using that variable of temperature. So the null hypothesis would be that temperature will have no effect on photosynthetic rate. The alternative hypothesis is that photosynthetic rate will be lowest when the temperature is lowest and highest when the temperature is highest. Now, that may not be a correct hypothesis because we remember from that data that super high temperature actually denatured the photosynthetic enzymes, and so the rate actually was lower. And that is reflected in the prediction. So the photosynthetic rate will be lowest with lower temperature due to a lack of kinetic energy. The rate will be highest at a temperature that is optimum for the enzymes used in photosynthesis. And that's because too high temperatures will stop the reaction by denaturing the enzymes, and the rate will actually fall to 0. All right, really quickly, just the task verb justify. In this question, students were given a bunch of background information that you can read in that first bullet point, and then you're asked to provide reasoning to justify this claim. So scientists claim that a plant has more than one type of pigment, and it would have a greater fitness than a plant that has only one type of photosynthetic pigment. And you're asked to justify this claim. And the response that you would have to justify this is that if you have more pigments, you would be able to absorb a greater range of wavelengths, and that should increase the plant's ability to perform photosynthesis and then survive and reproduce. Okay, so that gets in that evolutionary piece. Okay, so to debrief, today you should be able to describe the photosynthetic processes that allow organisms to capture and store energy. You should be able to explain how cells capture energy from light and transfer it to biological molecules for storage and use. And the key takeaway today is that highly complex organization of living systems requires a constant input of energy and the exchange of macromolecules. So I'm going to skip the common errors and misunderstandings right now in the interest of time. This is a former section of a free response question that has to do with bacteriorhodopsin, which is that type of pigment that's found in early photosynthetic systems. And I'm going to share with you the scoring guideline. And then there are some common errors and misunderstandings that you can read through when you read this on your own, because I'd like to move you forward and have you take a look at your homework for tonight. Okay, so go back and look at this and read it on your own. Okay, so here's your homework tonight, AP exam practice. In a certain prairie community, a dominant prairie grass species has recently been infected with a virus that disrupts one of the electron transport proteins in the chloroplast of infected cells. Describe the most likely effects on cellular processes. Be specific as to which processes and molecules are most likely to be directly affected. So make sure you talk about the cellular processes and then call out the molecules that are going to be affected. Okay, so one last word before I leave you today, and this is about your device and internet access. We are approaching our exam date, so if you need connectivity or mobile tools, please reach out to College Board and let us know. And the way to do that is through cb.org/tech. Please do this by April 24th, which is next Friday. Thank you so much for watching. I know today was some tough teaching and learning. I hope you'll join us again. Be sure to do your practice question. It'll get reviewed in the next lesson tomorrow, which is on cellular respiration. And I'll see you guys again next Tuesday. Have a great rest of your day.